Okay, then let's start locomotion and movement. So, what is locomotion? The locomotion is the movement of organism from one place to the another place. We say generally locomotion. It's a voluntary movement resulting in a change in location. That is must here. The locomotion is the movement of organism from voluntary movement of organism from one place to the another place. So what are the types of movements here? In general, we say amoeboid movement, ciliary movement, muscular movement. You can say amoeboid movement is generally performed by pseudopodia and the macrophages, leukocytes, cytoskeletal elements like microfilaments. These are all showing amoeboid movements. Next, the ciliary movements. Ciliary movements you can see in trachea, isn't it? In the fallopian tube, that means in the female reproductive tract, you can see the movements of cilia. Then the last one, the bigger one, that is muscular movement, which is done by muscle. The movement of limbs, tongue, jaw, what do you say it as muscular movements. So let's see the human muscular system first of all to understand the scenario of muscle contraction. The muscle contraction is the area which include a lot of applications. It includes muscles which are mesodermal in origin. Human muscular system include muscles which are mesodermal in origin. The muscles constitute 40 to 50% of the body weight. The muscles have excitability, contractility, and extensibility and electricity. We have three types of muscles in general that are skeletal or skeletal muscles. We call them as striated or voluntary muscles. Visceral muscles, which are also said to be as non-striated or smooth muscles, and the cardiac muscles. So let's see one after another. That skeletal muscles here, that striations are present in them by the arrangement of thick and thin filaments regular, which we are going to see that in detail in the coming class. The voluntary muscles they are in our control, they are attached to the skeleton. Hence we call skeletal muscles. The blood supply wise, they have the rich vascularization, rich blood supply. These muscles undergo fatigue. When they work fast, they undergo fatigue. That how do they undergo fatigue? What are the biochemical reactions now we are going to learn? The muscle fiber here is multinucleate condition. That means syncytial condition you can see. The numerous mitochondria are present here. That mitochondria are numerous. When it comes to the visceral muscle or smooth muscle, striations are absent here. That means the arrangement of thick and thin filaments are in irregular manner. That regular manner makes striations. Here they are irregular, that's why you don't find any striation. Hence we call smooth muscles. Then involuntary muscle, they are not in our will or control. What do you say? Involuntary muscle. The found in visceral organs like alimentary canal, reproductive tract, and urinary bladder. Then poor blood supply, as they don't require much here. Their working style is slow. That non-fatigue, they're working slow. That means they work slow. That the fatigue also not can be expected there. Uninucleate condition, you see. So mitochondria are comparatively less with the earlier one. Cardiac muscle, striations present here like skeletal muscles. Involuntary, these are also the involuntary. The found only in heart valve, that rich blood supply, non-fatigue condition. They should not undergo fatigue. Isn't it? The cardiac muscles should not undergo fatigue. For a minute, 72 to 75 minutes, 72 to 75 times, the heartbeat should occur. In between the time, fatigue means there is a death of person, heart attack. Uninucleate condition you can see here. And the numerous mitochondria are present. And I think we discussed about this particular intercalated is present between the cardiac muscle fiber. The cardiac muscle fiber arranges like network. You see, between every two blue lines, one muscle fiber. Should understand that one muscle fiber connected to the another muscle fiber with the help of intercalated discs. Let's see the differences here. Just take a look on the particular differences of this particular area. Yes, skeletal muscles, we said striations are present. Striations are absent in case of visceral muscle. The cardiac muscles are with striations. So, striation wise, cardiac and skeletal muscle. Involuntary, visceral and cardiac muscles are involuntary. These combinations are going to be questioned. Then voluntary muscle, only skeletal muscles are voluntary. Okay. Next, the skeletal muscles are attached to the skeleton. Rich blood supply will find for both skeletal and cardiac. Should see this particular in the form of combination. We'll have the combination questions there. So that rich blood supply, that means both skeletal and cardiac. Poor blood supply, visceral. That undergo fatigue, only skeletal muscle. Visceral and cardiac muscle, you don't find any fatigue. Multinucleate condition, you'll find in case of skeletal muscle. In general, visceral and cardiac are uninucleate in condition. Numerous mitochondria will have the combination with cardiac and skeletal. Visceral muscles are going to be less mitochondria, and you should understand the names also. What means that skeletal? Why it is called skeletal? Because they attach to the bone. Striated, because arrangement of thick and thin filaments in regular manner. Voluntary, that means they are in our will or control in the CNS, central nervous system. In the same way, visceral, mostly present in the visceral organ. Smooth, why they call smooth? That means the arrangement of thick and thin filament is in irregular manner. Involuntary, not in our control. Then when it comes to the cardiac muscles, the cardiac, because they are present only in the wall of the heart. Involuntary, they are not in our control. Then these cardiac muscles are even called striated. Why we call them as striated muscles? Because they are not in our that arrangement of thick and thin filaments and regular condition. Okay. So now let's continue. Let's continue here. Let's continue the process. The structure of the striated muscle. Just look towards this particular diagram. Then we'll be back here after all learning the complete procedure of this particular skeletal muscle. The muscle contraction process. Now we are going in the skeletal muscle. Okay, so muscle contraction process. Now we are moving in skeletal muscles. So let me go for that and let me take the board for this to make the particular process to understand that clearly. We need to enter into this muscle contraction, need to learn in detail. So because this is the area, what do you say, that with great application. So that's why we'll go with onboard. Let us learn this particular procedure. So I'm coming from the beginning. Let's see here the transverse section of the muscle. First of all, I'm going to draw the transverse section of muscle. So muscle contraction process we are going for. So transverse section of muscle. In the transverse section, the muscle is surrounded by epimysium. Each muscle is surrounded by epimysium. That the dense fibrous connective tissue membrane we call epimysium. Then a muscle include many fascicles. Each muscle include many fascicles. We call these particular structures as fascicles. Each fascicle contain muscle fibers. These are the muscle fibers. So many are present, but I am drawing for our clearance just 3 to 4. That muscle fibers are present. Bundle of muscle fibers we even call fascicle. Okay. So each muscle fiber is surrounded by a membrane called endomysium. So let me show here. These are the endomysium. Okay. So these are all endomysium. That blue line I am going to draw. Okay, so then let's come from the here. The fascicle is surrounded by a membrane called perimysium. Membrane called perimysium. And each muscle fiber is surrounded by that membrane endomysium. Okay, so membranes wise, that epimysium which surrounds the muscle. Then each fascicle surrounded by perimysium. Individual muscle fiber surrounded by endomysium. Okay. So then from muscle fiber wise, if you come, the each muscle fiber surrounded by endomysium. Bundle of muscle fiber is called fascicle. Each fascicle itself surrounded by endomysium. All the fasciculae are surrounded by a irregular dense fibrous connective tissue sheath is called epimysium. From here, this muscle fiber, you need to bring out the muscle fiber. I am bringing muscle fiber out. For that, let us learn the structure of muscle fiber. Each muscle fiber is surrounded by that membrane called sarcolemma. Each muscle fiber surrounded by a membrane called sarcolemma. The cytoplasm in the muscle fiber is called sarcoplasm. Cytoplasm in the muscle fiber is called sarcoplasm. The endoplasmic reticulum here present that along the length you will find, but I am drawing somewhere else. That sarcoplasmic reticulum. This language is also going to be question for us. Sarcoplasmic reticulum. The mitochondria of muscle fiber is called sarcosome. The mitochondria of muscle fiber is said to be sarcosome. Then, as we said earlier, that this is a multinucleate condition. Nuclei are arranged at the periphery. Multinucleate condition is seen over, and this muscle fiber is having many myofibrils along the length of the muscle fiber will find many muscle fibers. These are the muscle fibers. Okay, these are the muscle fibers here. So we need to see a particular. What is this? Sorry, that this is itself muscle fiber. Muscle fiber. We have a small spell mistake here. That each this muscle fiber contain many myofibrils. This is the myofibril. So that in the muscle fiber, that membrane, the plasmalemma of muscle fiber is called sarcolemma. The cytoplasm is called sarcoplasm. Endoplasmic reticulum is considered as sarcoplasmic reticulum. And the mitochondria here called sarcosome, which is a frequent and regular question. Just focus there. Sarcosome. A muscle fiber include many myofibrils. Let's take a myofibril in microscope. If you take a myofibril in a microscope, that this particular myofibril is alternately arranged with the dark and light bands. That alternately arranged dark and light bands you can see here. We'll show the arrangement clearly, but let me draw like this first of all. The dark and light bands, that myofibril is with so many dark and light bands are present here. So like isotropic band is a light band. Anisotropic band we say dark band. Isotropic band, light band, anisotropic band, isotropic band, anisotropic band. Okay, in the center of each light band we'll find jet line, jet line, jet line. Okay, so likewise, between the jet lines, whatever present, between the successive jet lines, whatever there, that we term it as sarcomere. To reach toward the sarcomere, we just learned everything like this, sarcomere. So after identifying the sarcomere, we'll go back. So what is sarcomere? Present between the two successive jet lines. So what I mean here, what I want to explain, a myofibril is having many sarcomeres. To explain that, we'll go the detailed structure of sarcomere. But what is sarcomere? That between the successive jet lines, whatever present, that is called sarcomere. What is present? That we'll see. So from the beginning, when it comes here, let's come from the beginning. We started with a muscle. A muscle contains fascicle. A fascicle contains a muscle fiber. A fascicle contains a muscle fiber. A muscle fiber contains many myofibril. In a myofibril, we'll find many sarcomeres. Okay, so muscle means a bundle of fascicle. Fasciculi, plural. Singular one we say fascicle. Each fascicle contains many muscle fibers. Each muscle fiber contains many myofibrils. Many myofibrils. I am taking little bit time here. That is because when you Focus the class here that no need to learn. Directly you can answer all kind of questions from this particular area. So myofibril. Each myofibril is having many sarcomeres. Okay. So this is the issue here. We reach toward the sarcomere. Okay. Many sarcomeres are present in a myofibril. Many myofibril. Let me see here that many sarcomeres are present in a myofibril. Then many myofibrils are present in a muscle fiber. Then many muscle fibers are present in a fascicle. Many fasciculi include we call muscle. So exactly where we are, we need to find it out. Then why should we go for sarcomere? What is sarcomere? The structural and functional unit of muscle contraction. That will be here like this in this way. That question will come for us. Structural and functional unit of muscle contraction. Who is called the structural and functional unit of muscle contraction? Means that we'll get the answer, that is sarcomere. So that's why let's locate that particular sarcomere first of all. Then a particular sarcomere, a single sarcomere I'm going to draw now. That the center of I band, we said Z line, isn't it? The center of I band, we said Z line. So this is the I band here. Let me take. This is the I band and this is the I band. Okay, center of I band is with Z line. Next, need to go with A band. So I'm using that A band with another color I'm using here. So this is A band. Anisotropic band. We can say this as anisotropic band. Okay. So when it come from the back side, we'll have a question always that a sarcomere include. See here that in the center of I band we said there is a Z line. That means half of the I band, a full A band, and remaining half of the I band. That means half I band, A band, half I band is called a sarcomere. So that you need to remember here. That let me say again. I'll use another color. Half I band, then that half I band, then A band, then half I band. So here these are the series of alternately arranged dark and light bands you can see. Let me show this in the detailed manner. So let me here. This is I band. Then now let me go clearly here. This outside I am drawing here. This is I band. Okay, what next? What next? That is A band. This is A band. Next I band. Next A band. So likewise, we'll find a series of I bands and A bands. Okay, at this time, I'm not drawing any thick and thin filaments there. Just we need to have an idea that I band, center of I band with Z line, center of I band with Z line. So whatever is here, this is called sarcomere. This is said to be sarcomere. So now let us see the arrangement of thick and thin filaments in the case of sarcomere here now. Then thick filaments. Thick filaments. First of all, let me draw the thick filaments. I don't draw much here because the visualization is very important in this area. If you can visualize this, you can answer any kind of application questions from this particular area. Okay, so this is a thick filament. The thick filament. Where does the thick filaments are present? The thick filaments are present in A band. Simply we say. Okay, thick filaments are present in A band. So thin filaments. Let us see where does the thin filaments are present. Thin filaments are there in I band. Thin filaments are present in I band. Okay. Do they are present in A band? Yes. But except in one place, we need to locate that particular place. Who is that? H zone. That is called as H zone. So in the H zone, you don't find that particular the thin filaments. Okay. So in the center of H zone, you can see there is a line, thin line that is called M line. M line. So understanding this particular phenomena is very important here. First of all, what we said, we need to understand What is sarcomere? A sarcomere means half I band, then half I band, then full A band, half I band. Then arrangement of thick and thin filaments wise, we are talking about that thick filaments are present in the A band. Thick filaments are present in A band. Next, thin filaments. When it comes to the thin filaments, in I band and in A band also the thin filaments are present, but except H zone. Thin filaments are present in the A band, but except H zone. So this is A band, I and I. I means light band, we say isotropic band. So now we have some questions based on this particular area. What are they? Only thin filaments are present. Only thin filament, this area, I band. Only thick filaments are present in this area. That means H zone. Both thick and thin filaments are present in this area. That means A band except H zone. So for these kind of things, this kind of visualization is very important. Okay, so let me say again, I band contains only thin filaments. A band except H zone contains both thick and thin filaments. H zone contains only thick filaments. Only thick filaments. So then we have to see what is muscle contraction exactly. What is muscle contraction? Muscle contraction means that, let me draw that Z lines again. Then A band. So we said this is a thick filament. Thick filament in the A band I am going to draw. This is complete A band. Now, what is muscle contraction? The thin filaments need to be pulled towards the head zone. Thin filaments are going to be pulled towards the head zone. Okay, here like this. Muscle contraction means the thin filaments are going to be pulled towards the head zone. So who is going to pull this particular thin filaments towards the head zone? These thick filaments. The thick filaments are going to pull these thin filaments towards the head zone. Simply a single line. What is muscle contraction? Thin filaments are going to be pulled towards the head zone. Do the thin filaments are not there in head zone? No, that you can see. This is a normal state, sarcomere at normal status. Then sarcomere at fully contracted muscle. In the fully contracted muscle, the muscle in full contraction means that some calculations will be changed over. Only thin filaments are present as usual, I band, as usual I band. Only thick filaments are present in. There is no place only with thick filaments because head zone is also filled with thin filaments. Okay, so just you understand this area. That you can understand the muscle contraction totally. Okay, so the muscle contraction can be understood. What is exactly what is muscle contraction? There we can understand by looking toward this particular area. Let me show that particular diagram here. Let me rotate this. Then you just try to understand this particular phenomenon. Okay. Just by looking toward these two diagrams, there you can understand what will happen, what's going to be happen here. Let me show. This is my notes. I'll send it to you. Soon I'll send the copies of muscle, these particular diagrams completely explained. Okay. So to reach this particular area, let's make a small revision. As I said over, you see that particular pattern. The muscle include fascicle, fascicle include that muscle fiber include that myofibril. In myofibril, there are so many thick and thin filaments. Okay, so in the same way that you can see that muscle is surrounded by epimysium, it contains fascicles, and each fascicle is surrounded by perimysium, and in the fascicle there are muscle fibers, they are surrounded by endomysium. Okay, then that we pulled out one muscle fiber, we pulled here, that's our language, sarcolemma, syncytial condition, sarcoplasm, and sarcoplasmic reticulum, sarcosome, that all we are showing here, and taking one particular diagram here. Let me erase this. that one myofibril is pulled out and showing that arrangement of thick and thin filaments in alternate manner. That I band and A band, you can see here clearly that I band and A band. Center of I band is with Z line. Center of I band, Z line. Center of I band, Z line. Center of A band is with H zone. Center of H zone, M line. So there you can understand here this particular pattern. Then what is sarcomere? Present between the two successive Z lines. Whatever present, that we say sarcomere. What next? What next here? So showing one particular sarcomere for you, that as I shown that you in color. Now that written diagram we are moving for. So this is the Z line. This is Z line. Then arrangement of thick and thin filaments, you can see here. So just this, you have to watch this particular difference between these two. And we have so many questions in this particular area. So the first diagram, whatever you are looking here, that is showing that arrangement of thick and thin filaments in regular manner. Then what is muscle contraction? Pulling thin filaments towards the head zone. Just pulling like this towards the head zone. When you pull the thin filaments, that automatically you see that the Z line is also getting down. So during this particular procedure, we have some questions. In the maximally contracted muscle, what about the length of I band? You see this dots, you can understand. Length of I band decrease, isn't it? The length of I band is decreased. Okay. So what about the length of thin filament? Anything happened to the thin filament length? No. Just it pulled towards the head zone. Length of thin filament constant. Length of thick filament constant. Length of I band decreases. Okay, so length of A band, nothing happened. Just see that particular dotted line there, you can understand. Nothing happened to the A band. Only the change is happening is thin filaments are moved towards the head zone. So that's why we say in the maximally contracted muscle that the particular H zone is going to be disappear. Head zone is going to be disappear. Okay. So for that, we'll watch a video here in this particular to understand. Then we get back into this particular scenario again. A small video will go for on muscle contraction. Then we'll get back to this particular. So I'm taking time here for the explanation of muscle contraction because this is the area with the very important and great applications we have in this particular area. Let's watch that particular video.
During contraction of a muscle, calcium ions bind to troponin. This moves tropomyosin out of the way and uncovers binding sites for myosin on the actin myofilaments. ADP and phosphate are attached to the myosin head in the previous cycle of movement. The myosin head attached.
Let me here. Muscle contraction, very fine muscle contraction. Yes. Just let's watch the muscle contraction.
During contraction of a muscle, calcium ions bind to troponin. This moves tropomyosin out of the way and uncovers binding sites for myosin on the actin myofilaments. ADP and phosphate are attached to the myosin head from the previous cycle of movement. The myosin heads attach to the exposed binding sites on the actin myofilaments to form cross bridges, and the phosphate is released. Energy stored in the head of the myosin myofilament is used to move the head. This causes the actin myofilament to slide past the myosin myofilament. The ADP is released from the myosin head as it moves. The bond between actin and the myosin head is broken when an ATP molecule binds to the myosin head. The ATP is broken down to ADP and phosphate, releasing energy, which is stored in the myosin head and will be used later for movement. The head of the myosin molecule returns to its upright position and is ready to bind to actin again. If calcium ions are still present, the entire sequence is repeated. During contraction of a muscle, calcium ions bind to troponin. This move
Yes, what we said, pulling thin filament towards the head zone is going to be muscle contraction. Here, we need to recognize first of all what are thin filaments and what are thick filaments. You see here, this is a thin filament. First of all, let us recognize this is a thin filament. You'll find over thin filament is having three elements. That violet color you are observing, that is actin, and the fibers which are surrounding that particular actin molecules are tropomyosin, and then you will find the some what do you say, red color, that is troponin. That means each thin filament is having three components: actin, tropomyosin, and troponin. Actin, tropomyosin, and troponin. So first of all, let us try to learn that particular elements of actin, troponin, and tropomyosin. Then after, we'll move back again that particular video. So that means the video explains us what we are going to do actually. As I said to you, that movement of the actin and troponin, tropomyosin along with the myosin. So the simply what I am saying, movement of thin filament towards the head zone. So to understand that phenomena, we need to understand that the biomolecular arrangement of the thin filaments and thick filaments. First of all, let me go for the thin filament here. Each thin filament is having three components: actin, troponin, tropomyosin. Let me show them individually. Actin is in the form of two, G-actin and F-actin, globular actin, individually like this. F-actin, if you take F-actin, that particular molecules are arranged in the linear series, we call them as F-actin. Okay, so we have worked with F-actin here, as you have seen in the video, that filamentous actin. Next, what is tropomyosin? That we are seeing the tropomyosin is a fiber like these particular fibers are present. Then troponin. Troponin is always available in the form of troponin, in general called TN, is always available in the form of three grouped molecules. This is troponin. Okay. Let us arrange all the three in an actin filament. Actin filament here. Let us arrange all these particular things in actin filament. Sorry, what do you say? The thin filament. That how does the thin filament is getting arranged? Let us see in the thin filament. Okay, so first of all, I am going to draw the actin molecules here. Actin molecules. Then the troponin. You see, this is the troponin. The troponin surrounds that actin molecule. So where they are surrounding, where exactly active sites are present, where exactly the active sites are present, there that tropomyosin is surrounding that particular. Next, we need to recognize that troponin molecules. So wherever that particular tropomyosin is covering the active site, there you can see the troponin molecule. Okay, so in that way, the troponin which attaches to the tropomyosin is called as TNT. The troponin which is inhibiting the active site not to expose by supporting TNT is called TNI. The troponin which can bind with the calcium ion is called TNC. So everywhere is the same manner: TNT, TNI, TNC, TNT, TNI, TNC. So what exactly is happening here? The active site. What is the meaning of active site? The green one I draw. That particular myosin head should bind here and should pull the thin filament towards the head zone, which actually in the normal condition that active sites are covered by tropomyosin, they are supported by the troponin molecules. This is from thin filament side. What is the requirement from the thin filament side? The thin filaments, the active sites need to be exposed. How? Whenever the calcium ions will come and bind with the TNC, whenever the calcium ions will come and bind with the TNC, this particular TNC lift to the TNT for the exposure of active sites. I will show here a little bit sliding of the tropomyosin you can see with these particular molecules. So who is getting exposed over? Active sites are exposed. So see the diagram difference. In first diagram in normal condition, you can see that the active sites of the actin is covered by tropomyosin. So whenever the calcium ions will come and bind with the TNC, that lift the tropomyosin, that slide the tropomyosin, and now the active sites are exposed. So from this side it's over. The same way need to see the myosin head side, myosin molecule. So where we are exactly we need to see first of all. So let me show you. As I said, there are thick filaments and thin filaments. Thick filament and thin filament. Now I completed this one. So let me show this particular here. That whatever we drawn, these are the actin molecules, and they are covered by tropomyosin. And where exactly the active sites are present, there TNT, TNI, TNC are present. Okay, so you should understand like this. Now we are moving with the thick filament, which is nothing but called myosin. Structure of myosin we need to go through. So now I am closing here. That in the next, immediately just log it out. We'll complete the muscle contraction today. That I'm closing this and log in immediately to continue the muscle contraction.